Hello. My name is Kathy Ivey, and I'm a cardiovascular and stem cell researcher, and I'm excited to share with you today how Gladstone researchers are using gene editing to engineer human cellular models for the purpose of studying disease and developing therapeutics. As many of you know, 10 years ago, Shinya Yamanaka, Gladstone investigator, reported the amazing discovery and development of something that he called iPS cells, or induced pluripotent stem cells. These offered a huge advance over the use of human embryonic stem cells in research for several reasons. But what Shinya did to develop iPS cells is that he showed that a researcher could take the skin or the blood, any cell really, from an individual and reprogram those cells to a pluripotent state, much like an embryonic stem cell, through the activation of genes that define that embryonic stem cell state. And then these resulting iPS cells would be nearly indistinguishable from embryonic stem cells in that they would renew infinitely, and they have the potential to become any of the 200 or so types of cells within our body, so cells of the heart, of the brain, of the gut. So this was amazing in that it overcame a lot of the ethical issues that were problematic in human embryonic stem cell research, but it also opened up amazing new avenues for just in science for researchers in that in the first time it allowed us to create really meaningful models of human disease, development and disease, and gave us the opportunity to move away from some of the classic models of non-human cells, which were used for discovery and drug development, and away from rodents, which make great models but differ in their physiology from that of humans, and gave us a chance to really study healthy or diseased cells from the human population. And so as a cardiovascular researcher, I've spent many years doing disease modeling for the heart and using iPS cells to do that. One of the things that is pretty remarkable in disease modeling is we want to study diseases often that are genetically predetermined. And we heard from Sandy that our genome is made up of about 3 billion nucleotides. And disease, cardiovascular disease or otherwise, can arise from a mutation or a variation in as few as one of these 3 billion nucleotides in our genome. So we were modeling those types of diseases in a dish, initially by taking skin cells from a person with disease, converting them into iPS cells, and then making heart cells from them so we could study the disease in a dish. And of course, we need a control population to know what normal, healthy heart cells would look like. So, and we would like that sample to be genetically similar to that of the diseased individual. So we would take, for instance, a sample from a healthy relative, maybe a mother or a daughter of the individual whose disease we were studying. And that was great. It was a pretty good match, but it's important to remember that in addition to that one nucleotide that we want to study that's different between that mother and daughter, there's another about 1.5 billion nucleotides that differ between those two individuals because they only share, in actuality, about half of their genome. So this is not ideal for a scientist or an ideal way to get the most meaningful data because we're not comparing the perfect control in our disease studies. So what we wanted to be able to do was to make that single nucleotide mutation in the background of a healthy person's genome. And this is just an example of how, as scientists, no matter how great our tools are, and we were gifted with this amazing technology of iPS cells, we want to make our tools better to give more meaningful data. So you heard also that as scientists, we tend to learn from nature. When we have a problem in molecular biology in particular, we will take time to observe how nature solves that problem and what are the tools that nature has created to solve that problem, and then we'll try and borrow that or steal that from nature for our use in the laboratory. And so that brings me to CRISPR for genome editing. Can I see a show of hands from people who have heard about CRISPR in the news lately and gene editing? Fantastic. So you heard a little bit about CRISPR already. I'll remind you what it really boils down to is that in nature, CRISPR-Cas9 is a bacterial defense system. So bacteria use this as a way to defend themselves against viral infection. We already heard about viruses from Lior and the fact that they contain certain DNA sequences. So in bacteria, Cas9, which is the protein component of the CRISPR-Cas9 system, uses DNA sequences to identify viruses as foreign and then cuts that viral DNA to inactivate it. And so researchers who were studying this system saw a lot of potential in this, and specifically for capturing it for the use of human gene editing in human cells. And so they tried borrowing that CRISPR-Cas9 system from bacterial cells and applying it to human cells. And amazingly, what they found was that Cas9 could be directed to specific sites in the human genome based just on sequence. So by giving the Cas9 protein the right sequence to target, it can look through those 3 billion bases and find the exact sequence that we'd like to target it to. And when it finds that sequence, it cuts it. And in human cells, when DNA is cut, that the cell mounts a DNA repair response to fix that cut DNA, and that's essential for our long-term survival as humans. So researchers have taken advantage of these two things, and we're making site-specific cuts in human DNA and human cells, and then supplying a template for the cell to use when it's carrying out that DNA repair that it was already going to carry out. And in doing so, we're able to introduce specific DNA sequences exactly where we want them in the human genome. And that's really the basis of CRISPR-Cas9 in human genome editing. So that's exactly how Gladstone researchers are going about making better disease models. We're able to do now what we wanted to do for a long time, making these ideal disease models. So using CRISPR, we can edit disease-causing mutations into healthy human iPS cells, and then compare heart cells or brain cells or gut cells that are made from those healthy or diseased iPS cells, and then isolate the exact effect of these single disease-causing nucleotide changes out of the 3 billion. And so this has allowed us to uncover cellular and molecular mechanisms of disease that were hidden before in our previous studies. But that's not all that CRISPR editing is good for. It's being used for a whole lot of things. Some things that Gladstone researchers are doing here with CRISPR-Cas9 genome editing is engineering lineage reporters. So I told you that iPS cells can become any of the 200 or so types of cells in our body, and they do that in the laboratory all the time, but it's a difficult thing to observe. So as a cell becomes a heart cell, it's difficult to observe that happening. They don't send out a flare letting us know that that is happening, but we can engineer them to send out a flare. So using CRISPR-Cas9, we can edit in the code for example, for a green fluorescent protein, so that when a cell adopts a certain lineage, that of a heart cell or a brain cell, it will activate this green fluorescent protein and give the researcher a signal, hello, I'm becoming this cell, capture me and study me. Gladstone researchers are also engineering tags into genes so that when that gene is made into the protein product, that's the machine of the cell, the protein becomes labeled in a way that's very useful for tracking it, detecting it, and capturing it, and understanding its location and activity. And that is really what's enabling us to get at these cellular and molecular mechanisms of disease. And then researchers are also using clever variations on CRISPR-Cas9 technology to either activate or inactivate specific genes in living cells. And that allows, as Sandy was talking about, the adoption of specific cell fates. So if you're interested in talking more about engineering human stem cells for research purposes, or you'd like to meet with one of our scientists who's doing this in the laboratory now, I'd be very interested in talking with you at the reception. Thank you.